Hi, everyone. It is my great honor today to join today's LabRule webinar to share some of our recent studies on the PD-L1 glycosylation and the cancer immunotherapy. Hopefully, through this presentation, we will have an idea how we translate basic research to the clinical use in a hope of benefit patient at the bedside. Cancer immunotherapy has drastically reshaped the landscape of cancer treatment since the development of anti-CTLA-4 antibody in treating myeloma in 2010. Today, there are over 500 clinical trials. Total of 9 types of monoclonal antibody developed by a pharmaceutical company are currently tested at the clinical site in treating 20 types of cancer on over 20,000 patients. So today, our objective is to first understand the new challenge of breast cancer therapy. Our lab has tremendous interest in studying this type of disease for 2 decades. We have also like to take this opportunity to advocate that there is a special need for the breast cancer patient to be breast cancer research to be continually supported. Because the breast cancer mortality, it wasn't like 20 or 30 years ago. Breast cancer was so devastating. Since the development of hormone therapy or target therapy, the mortality of breast cancer has significantly reduced. So now the 5-year survival rate is over 90%. So indicate that in past 10, 20 years, scientists do achieve a great advance on treating this type of disease. However, this disease still have a lot of issues such as recurrent metastasis or drug resistance. So therefore, we still would like to take this opportunity to continuously advocate for the patient that there is a tremendous need for federal government, for philanthropists, for basic research to focus on this type of disease. And second, we would like to introduce what we have been finding in the past 3 years. We focus on the glycosylation of PD-L1, and we translate this study to immunotherapy strategy development. So we would also touch base on that to tell you how we identify from of this important regulatory mechanism. And lastly, I would like to briefly introduce some of the new technology we developed in the past 3 years. By working with the SM Bioscience, we developed several state-of-art type of methodology to try to help us to understand about basic science and try to translate this research to clinical use. First, breast cancer, as I just mentioned, has been tremendous treated in the past 20 years. So, however, American Cancer Society still estimate that in the 2016, the new case identified in the woman, there will be 29% of the patient will be identified as the breast cancer. So this suggests that although the mortality has dramatically reduced for this disease, however, the incidence of this disease is still very high. So therefore, we as a basic scientist, we still have a lot of room to try to improve the treatment or the quality of the breast cancer patients. One type of breast cancer of our interest is called triple-negative breast cancer. This subtype of breast cancer lacks of estrogen receptor, progesterone receptor, and the human epidermal growth factor receptor 2. Because this type of disease lack of the treatment targets such as above mentioned the 3 molecules, so therefore there is basically no treatment for this type of disease. Patient diagnosed with triple negative usually goes through the traditional chemotherapy. Unfortunately, only half of the patients can benefit from this therapy. which means another half of triple-negative breast cancer has basically no drug to cure. So to our interesting, we found out that among all breast cancer subtypes, triple-negative breast cancer has the most abundant tumor-infiltrated lymphocyte. This lymphocyte present in the tumor supposed to help us against the cancer growth. But somehow this triple-negative breast cancer has the mechanism to silence, to inactivate this lymphocyte inside the tumor area. So this also gives us a hint that in the triple-negative breast cancer, if we use the immunotherapy to reactivate the tumor site lymphocyte activity, will be a new therapeutic strategy to enhance the triple-negative therapy. Today, one of the most important immunosuppression mechanisms has been identified. It's called PD-L1 and the PD-1 signaling axis. So the next slide showing you is a working mechanism of this molecule. So as you can see on the panel A, the cartoon shows that the tumor cell has expressed a lot of tumor antigen. It's exposed by the NHC1 class on the cell surface. And this presentation, once the T cell recognize this presentation, T cell will come into the tumor area and try to eliminate this cancer cell rapidly. However, cancer cell is also very smart. They will express the PD-L1 on the cell surface. This expression will engage with the T cell on the PD-1, and this engagement will eventually shut off the T cell activity, cause immunoescape. Scientists now have developed monoclonal antibody that can specifically recognize either PD-L1 on the cancer cell or PD-1 on the T cell. This type of strategy will help to mask the interaction surface of these two molecules and therefore block this PD-L1 and the PD-1 engagement in an effort to reactivate the T cell activity, allowing T cell to recognize the cancer cell and then eliminate this foreign substance. Today, this type of immunotherapy has reached a tremendous improvement, focused mostly on the melanoma, Hodgkin's lymphoma, lung cancer, kidney cancer, and bladder cancer. And there are 4 drugs that have been approved by the FDA currently is available, and there are another 5 drugs that is under clinical trial right now, all targeting on the PD-L1 and the PD-1 signaling axis. Then the overall response rate of this type of treatment is around 20 to 40%. Common side effects including diarrhea, joint pain, autoimmune disease have been found, but those are mild and can be managed by steroid. And right now, there are more and more questions such as intrinsic and acquired resistance, hyperprogressive disease, and try to identify new biomarkers to predict the response of the anti-PD-L1 and PD-1 is a new challenge that awaits us to study. So as I mentioned earlier, triple negative has inside the triple negative breast cancer, there is a lot of infiltrated T cell, and however, cancer cells still grow. So therefore, we suspect that triple negative breast cancer must have some kind of immunosuppression mechanism. So one molecule of interest is PD-L1. This protein is expressed on the cancer cell, and is supposed to have molecular weight 33 kilodalton. So as a cancer biologist, what we first like to do is to monitor, to test where does the PD-L1 express in the cancer cell line. So in the panel A, we can see that across breast cancer, melanoma, lung cancer, and the colon cancer cell lysate, we did a simple Western blot. We tried to recapitulate the PD-L1 expression on the Western blot, and we found that this protein expression at the molecular weight 33 is very little. So we have tried several ways to enrich this expression, but it was not very successful. So in lifetime, we actually, interestingly, we noticed above the molecular weight 50 kilodalton, there is a huge abundant, we saw non-specific expression of the PD-L1 because the molecular weight is totally wrong. And it expressed as a heterogeneous expression. It's not a distinct pattern that we used to expect on the regular Western blot. So we didn't pay much attention in lifetime till we use the overexpression system. This experiment we did is we cloned 33 kilodalton PD-L1, and we expressed this protein in the 293 cells. We found out, again, there is no 33 kilodalton protein expression on the Western blot. Instead, there is a 50 kilodalton molecular weight of PD-L1 expression. We made several contrasts, and they all seem to act similarly. So therefore, we start to suspect whether this 50 kilodalton molecular weight of the protein of the signal is actually the PD-L1. To confirm this result, we did two shRNA knockdown clone, knock down the endogenous PD-L1, and we found out the 50 molecular weight kilodalton signal was disappeared. When we reconstitute the wild-type PD-L1 into those knockdown clones, we found out the molecular weight 50 kilodalton signal was reappeared. We also create another two CRISPR-Cas9 knockout of PD-L1, and we consistently found out that the molecular weight 50 kilodalton is disappeared. So this result suggests that this high molecular weight of the protein is real PD-L1 signal. Again, like I earlier mentioned, our lab has tremendous interest in studying triple-negative breast cancer. So what does this PD-L1 express in the breast cancer? In this slide, we also use the panel of breast cancer cell line, and we group them into non-triple negative and the triple negative breast cancer cell line. And we did a simple Western blot with EGFR and the PD-L1. EGFR is highly expressed in the triple negative breast cancer, which has been known. And the PD-L1 expression also show very high abundant expression at the molecular weight 50, whereas the molecular weight 33 kilodalton here in the red arrow, we don't see much expression. Then we look for the literature to see what might be the reason cause the molecular weight shift of the PD-L1. So one thing come to our mind is that the protein glycosylation might cause the cell protein molecular weight migration dramatically. So protein glycosylation is attachment of sugar moiety on the protein to the glycoprotein. It usually occurs approximately predominantly at the NXT motif. N stands for asparagine, X is any amino acid, and the T is threonine. Glycosylation usually happens in the ER and the Golgi apparatus. And it is estimated that approximately 1/3 of all human protein has this type of modification. They are either N-link, O-link, or C-link glycosylation. Glycosylation controls protein folding, localization, interaction, and enzyme activity. So next, we would like to know whether what we observed, molecular weight 50 of the PD-L1 is really through glycosylation. So in this experiment we did we overexpress different tags of the PD-L1. In this case, we have MYC tag at C-terminal, we have flat tag at C-terminal, and we create another HA tag at N-terminal. We overexpressing this construct and then treat the cell with tunicamycin. Tunicamycin is a type of chemical that can block N-linked glycosylation synthesis. So we found out when we increase the concentration of tunicamycin, the molecular weight 50 of the PD-L1 start to decrease to molecular weight 33 kilodalton. So this result suggests the PD-L1 is a highly glycosylated protein, regarding as long as we use an overexpression system or when we look at the endogenous level. So next, we would like to know where does this glycosylation happen. So next, we do we check the mouse PD-L1 and the human PD-L1 sequence. We found out there are 4 NXT motifs consensusly found out in the extracellular domain of the PD-L1. They are N35, N192, N200, and N219. So therefore, to understand which site involved in this glycosylation event, we first create single mutation on the PD-L1. And we consistently see that all 4 single mutations has ability to reduce the protein migration of the PD-L1. So then we continuously create double mutant and a triple mutant. PD-L1 migration was continuously reduced till we create a 4-1Q. When we complete mutate 4 glycosylation sites, we found out PD-L1 expression goes to molecular weight 33 kilodalton. And as I showed you earlier, that wild-type PD-L1 treated with tunicamycin also can reduce to non-glycosylation form. So in this case, we can see a nice side-by-side together with these 2 molecules, which suggests that PD-L1 is only glycosylated at these 4 sites. We also create another 3 asparagine mutant at the non-NXT motif, and they do not change any molecular weight shift of the PD-L1. So on the left side, we did a mass spectrum analysis, collaborate with the academic Sinica in Taiwan. We identify this N35, the glycan structure of this N35 PD-L1. Of course, we also identify the other glycan structure, and because of the space issue, we are not going to show you here today. Since PD-L1 is highly glycosylated, so the next we would like to know is whether this bulk glycan structure will affect its interaction with PD-1. To do this, we first set out an in vitro binding system. In this case, we overexpression flag tag PD-L1 in the cell, and we use a flag bead to pull down this flag tag PD-L1, and then incubate with PD-1 protein in the solution. And after that, we did a pull down experiment. We found out that the wild type PD-L1 can interact with PD-1 efficiently. However, when non-glycosylated PD-L1 do not interact with PD-1. To test this hypothesis in vivo, we use a InQSI lifetime imaging system to quantify the dynamic change between PD-L1 and PD-1. So in the first picture we show in the red color is BT549 cells has nuclear IP expression. And then this cell will also express a wild-type PD-L1, which is a glycosylated form of the PD-L1. And then we add a fluorescence-labeled PD-1 protein in the solution. After 12 hours of time, we can measure the dynamic interaction between PD-L1 and the PD-1. So as you can see that there is a huge accumulation of red color on the cell surface after 12 hours of treatment. And on the other hand, in the non-glycosylated PD-L1 stable clone, we don't see any much green fluorescence, suggests PD-L1 and PD-1 do not interact after 12-hour period. So here we show you 3 individual clones. They all act similarly, which consistently support with our in vitro data show that this glycan structure is required for protein-protein interaction. So to understand the function of this the glycosylation, we did a T-cell killing assay. So in this experiment, again, we use an InQSI machine to mimic the dynamic interaction, the cell growth of the cancer cell and in the presence of T cell. So in this case, it's a BT549 nuclear IP clone, and we add a activated T cell, incubate together for about 5 days of time, and we can see the green fluorescence, which indicate the caspase-3 activity. As soon as the cancer cells start to die, we will see the reduce of the red color and also the gain of the green fluorescence. And this quantification tells us that indeed glycosylated PD-L1 cancer cell has the ability to resist to T cell killing, whereas non-glycosylated is very sensitive to T cell killing. Next, we performed the in vivo experiment to confirm this result by inoculate 41 PD-L1 wild type and the PD-L1 non-glycosylated PD-L1. And then into the biopsy mouse, we found out at 2 weeks of time, there is a huge geometrically gross differences between these 2 clones, suggests that loss of the glycosylation, the cancer cell loss of its ability to suppress T cell activity. And we also did a functional analysis, try to understand what's the difference between glycosylated form and the non-glycosylated PD-L1. So in this case, we do the cyclophosphamide post-stress experiment. We found that the wild-type glycosylated PD-L1 is very stable, whereas non-glycosylated PD-L1, the half-life is very short. And the 2 systems all indicate the same phenomenon. So next, we would like to know what's the upstream signaling that regulates this glycosylation event. So what we did is we took 2 triple-negative breast cancer cells, BT549 and 468 cells, and we treat with the different stimulator such as EGF, IGF, HGF, FGF, and the TGF-beta. We found consistently EGF can induce PD-L1 glycosylation in 2 triple-negative breast cancer. Because EGF is a ligand of EGFR, so we also confirm this result by adding different type of EGFR ligand. And we found that as long as EGFR being activated, the PD-L1 expression also increased. And the right panel shows you the flow cytometry measure the membrane bound PD-L1 is being upregulated by EGF ligand. So lastly, we would like to use this model to conclude our finding that EGFR has the ability to stabilize PD-L1 because non-glycosylated PD-L1 will be recognized by the GSK3 beta and thereby recruit the beta TRCP to to degrade this PD-L1 protein. Once PD-L1 get glycosylated, it will prevent the GSK3 beta recognize the PD-L1 and therefore cause the immunosuppressive activity. And lastly, since our signaling analysis indicate that EGFR is one of the key signaling that induce PD-L1 glycosylation, we suspect that the EGFR, if we use a tyrosine kinase inhibitor to inhibit EGFR, will be able to downregulate PD-L1 expression in the cancer cells. So here is the flow chart that we perform this animal analysis by treating every day daily with a Genevapin, and also every 3 days we treat with 100 micrograms of anti-PD-1 antibody. And we found out the combination of these two therapies significantly reduce the tumor growth of the 41 mouse cells. And this mouse has increased the mouse survival about 20% by combining these two therapies. We also look at the TIL analysis inside the tumor area, which is CD3, CD8 positive. We look at the interferon gamma expression, and consistently combining these two therapies enhance dramatically interferon gamma secretion. And also we look at the granzyme activity, found out that the tumor area that when we combine these two treatments, the granzyme activity of the cancer cells can be activated. So conclusion of this, the first part of my talk is that we found PD-L1 is unlinked glycosylated at 4-NXT motif. GSK3 beta can phosphorylate and destabilize non-glycosylated PD-L1. And glycosylation can stabilize PD-L1 through prevent GSK3 beta binding to non-glycosylated PD-L1. And the upstream signaling is the EGF that can increase the non-glycosylated form of PD-L1. So if we combine you inhibit the EGFR signaling, we are able to sensitize cancer triple-negative breast cancer cell to anti-PD-1 therapy in the syngeneic animal model. Next, I would like to quickly switch gear to show you one of our another finding. We also incorporate a lot of methodology using the SM Bioscience Intusight microscope, and we have published another paper in the Cancer Cell 2016. So what we found is that triple negative will also induce chronic inflammation. So we would like to know what's the inflammation, why this inflammation can cause cancer progression. So we found that inside the tumor microenvironment, macrophage has the ability to secrete the TNF-alpha, and this TNF-alpha will recognize cancer cell TNF-alpha receptor. So in this enlarged cartoon on the right side, the underlying mechanism is that the TNF-alpha will activate NF-kappa B and cause the P65 nuclear translocation, and thereby turn on the CSN5 expression. And we identify that CSN5 is a novel deubiquitination enzyme, and this enzyme will stabilize PD-L1 and therefore translocate, induce the cancer cell immunosuppression function. So in the end, we found in this study that in the tumor microenvironment, the 3 cell, 2 immune cell, macrophage and the T cell, and how these 3 cells interact together in the tumor microenvironment related to the immunosuppression activity. Next, I would like to spend some time to also discuss our recent study that we work with the ST-Cube Pharmaceuticals. We try to identify the first anti-glycosylated antibody. As we showed you earlier, that triple negative has very high abundant expression of the glycosylated PD-L1. So we are wondering if we can generate antibody that can specifically target this fold structure of the PD-L1, we may generate more therapeutic impact. So in this regard, what we did is we inoculate glycosylated PD-L1 as an antigen into the mouse, and then we create 3,000 hybridoma clone. So then next, we use a flow cytometry to screen those 3,000 clones, try to look for the antibody that can specifically recognize glycosylated PD-L1, but not non-glycosylated PD-L1. And then we use the InQSight machine to help us to further narrow down which antibody has the ability to neutralize PD-L1 and the PD-1 interaction. We finally narrow down 2 monoclonal antibody that has the ability. So this is a slide showing you that STM10004 and the STM108 antibody. These 2 antibodies has a very profound effect in inhibiting PD-L1 and the PD-1 interaction. So next, we would like to know where does these 2 antibodies bind to the PD-L1 glycan structure. So in this case, we create 3 NXT motif mutation, and we keep 1, in this case is N35 as a wild type. And we keep individual glycan cell, glycan structure as a wild type, and then we did a simple Western blot. We found out that STM108 antibody can specifically recognize N192 and N200. And based on our earlier mass spectrum analysis, we already know that there is a very unique glycan structure appear on this N192 and N200, which are the 2 sites that can be recognized by our STM108 antibody. Because earlier studies show that glycosylation of the HER2 has the ability to cross-link the membrane-bound HER2 and therefore induce clathrin-dependent HER2 internalization. So this paper helps us to hypothesize that whether this our antibody, anti-glycosylated PD-L1 antibody, also have the ability to cross-link a membrane-bound PD-L1 and therefore induce this PD-L1 internalization and goes to the lysosome and further get degraded in the cells. So to do this, we incorporate with SM Bioscience technology and also with the pH Rodo system. To do this, we fluorescence label our STM108 antibody with a fluorescence dye. And once this, so the theory is that when the cells start to uptake this antibody, the pH value will start from the membrane, which is pH 7, drop to the pH 4.5 inside the lysosome. And this acidic condition will help the linker region of the antibody to get cleaved. Once this linker region get cleaved, it will illuminate the red fluorescence in the microscope. So in this case, we are able to use the InQSight lifetime imaging system to measure the fluorescence, red fluorescence, and also in the real time, we can take the picture how and where the red fluorescence appear. So in this case, we can see in 12 hours period of time, when we add the STM108 antibody into the BT509 stable clone, we can see very nice red fluorescence induced, suggest that STM108 antibody can specifically internalize into the cancer cell. And once it internalized, we can see we did a Western blot, and this was done in the after 5 days of treatment. We can see that significantly amount of the PD-L1 is reduced suggests that this antibody has the ability to internalize PD-L1 and further degrade PD-L1. So why this internalization and degradation is so important? Because recently there is a antibody drug conjugation technology that is available. So we are thinking whether we can take this advantage to conjugate cytotoxicity drug into our antibody. So in this case, we choose the NNAE as our warhead, and we conjugate in our conjugation. On average, there are 4 chemo drugs being conjugated on our antibody. So we test the efficacy of this antibody in the vitro and also in vivo. We found out that only the cell has PD-L1 expression can specifically being treated by this antibody. This is in vivo, and here, when the mice has the PD-L1 expression, their tumor growth after treatment with our ADC is dramatically reduced. So this is the 41 human PD-L1 stable clone. They do not grow after we treat with our glycosylated ADC antibody. So this result is very exciting. So we also look at the tumor microenvironment to see that using the fluorescence IHC staining to detect the activated caspase-3 activity. So in this case, in the red color, indicate the caspase-3 activity. When the mice treated with glycosylated NNAE antibody, we can create a dramatic caspase-3 activation in the tumor microarea. We also did a cytoph analysis. We found that mice treated with the glycosylated NNAE antibody has dramatically tumor cell reduction, but slightly the immune cell get infected. So this result suggests that this antibody can selectively target the cancer cell, but not the immune cell, because as I mentioned earlier, that glycosylated of PD-L1 is highly abundant in the triple-negative breast cancer cells. So lastly, I would like to use this model to summarize this part of our study, which is that EGFR is the major signaling that can induce some sort of glycotransferase induction. And this glycotransferase will further glycosylate the PD-L1, and then this PD-L1 goes to the cell membrane. If we can create an antibody that specifically recognize glycosylated PD-L1, we'll have ability to induce this PD-L1 protein, and also once this endocytosis event happens, it will go to the lysosome and then get degraded. Once the antibody and the PD-L1 get degraded, the cancer cell cytotoxicity drug will be released, and then to kill the PD-L1 high expression cell, and also create a bystander effect, which will eradicate the cancer cell that do not express PD-L1. So this will create a profound therapeutic effect in reducing, eradicating triple-negative breast cancer. So the conclusion, what we found throughout this study, is that glycosylation is a very complex structure, and it's very difficult to assess in the regular basic research lab. So in this regard, glycosylation is often being neglected. So based on our signaling analysis, we can design a strategy and then try to isolate monoclonal antibody that can specifically recognize glycosylated PD-L1. And we first found out that this PD-L1 can cross-link membrane-bound PD-L1. So PD-L1 antibody can cross-link PD-L1 molecule and therefore induce the endocytosis of this protein. And through this endocytosis, we can let the antibody to carry a cytotoxicity drug and then induce a profound therapeutic impact to triple-negative breast cancer.